In this video, we're going to talk about the role of B cells in the immune response and how they work to help fight infections. As you can see on this slide, when a B cell becomes activated by binding to a receptor antigen complex binding and forming this complex, it causes cloning of the B cells, which means more B cells are produced that will specifically respond to the antigen or the pathogen that initially caused the activation. In the case of these cloned B cells, some of them are going to become plasma cells, cells that are capable of producing antibodies, and some of them become memory cells, which are going to be responsible for providing immunity or resistance to that particular pathogen in the future. So let's talk about the steps in antibody production. How do the B cells produce antibodies? So T cells and B cells don't work independent of one another. As you can see in the table here, which is table 16.6 in our textbook, the first step is the antigen-bearing agent, the pathogen, enters the tissue. When this happens, antigen-presenting cells like macrophages and subs engulf the cell through phagocytosis, present the antigens, and cause the T cells and B cells to become activated and then start producing the various different types of T cells and B cells that are going to be responsible for fighting the infection, including the plasma cells, which will eventually produce antibodies that can combine with the antigen that initially started the whole thing and help to fight the infection. So what is an antibody? An antibody, or an immunoglobulin, is a type of protein, plasma protein. And we talked a little bit about plasma proteins in the cardiovascular system structure. And while there are different kinds of antibodies, they all have some similarities. They all have a specific structure or basic structure that includes a constant region, which determines the type of antibody we're talking about, and a variable region, which determines which antigens that antibody will fight. And you can see the heavy chains and the light chains are also part of this structure. So there are 5 different classes of immunoglobulins or antibodies. IgG are the most common type of antibodies, and they're found both in plasma and in tissue fluid, and they work against a variety of various pathogens, including bacteria, viruses, and various toxins, and they play a role in activating complement, which is part of the non-specific defenses, which helps to destroy virally infected cells. IgA is found in glandular secretions. It's usually a dimer, and it acts against bacterial cells and viruses as well. IgM is a pentamer, or 5 of these antibodies combined into one large complex, and they're found primarily in the plasma. They're usually the first immunoglobulins that show up at the site of infection. IgD functions primarily as receptors, so most of our B cells have IgD on them, and they play an important role in activating B cells and causing them to differentiate into plasma cells and memory cells. And finally, IgE plays a role in inflammation, part of our non-specific defenses, but also can play a role in allergic reactions that we'll talk about in a later video. So what do antibodies do exactly? Well, there's 3 general ways that antibodies play a role in immunity: direct attack, activation of complement, and localized changes. So in direct attack, the antibody directly does something to the pathogen, to what contains the antigen, that causes it to no longer be harmful or causes it to be destroyed. And this can include agglutination, where antigens clump up. We saw an example of this when we did the blood typing reaction. Precipitation, which causes antigens that are normally dissolved in the fluid to become insoluble, which means then they can't enter the cells and cause problems. Or neutralization, where the antibody coats the antigen and causes it to lose its toxic properties. Complement, where you have antibodies and antigens combined together, activating complement. There are various effects that are involved in that. Optimization, where the cell membrane becomes more susceptible to being engulfed by the macrophages and other phagocytic cells. Chemotaxis, which means attracting the macrophages and neutrophils, the phagocytic cells, to the site of infection. Agglutination, which we've talked about already. Lysis, which where complement pokes holes in the foreign cell, causing the cell membrane to rupture and destroying the cell. And neutralization, again, in this case we're talking about viruses being neutralized, keeping them from being able to infect cells in our body. And finally, localized change is inflammation, and we talked about that. You have the 4 signs of inflammation. You have pain, you have redness, you have heat, and you have swelling. And inflammation, part of that is a walling off reaction, which helps slows down the spread of the pathogen, but also can speed up the repair process, speed up the reactions needed to help fight off the infection. So when you have an immune response, and specifically a humoral or an antibody or chemical immune response, there's 2 types of responses, I guess you could say, you have. You can have a primary response, which is what happens the very first time that you get exposed to a particular antigen or particular pathogen, and a secondary response, which happens if you get exposed to the same antigen a second or third or fourth time. As you can see from the graph here, the primary response takes longer to reach its maximum antibody level, and it doesn't ever get to as high of an antibody level. So in a primary immune response, remember, you get plasma cells, but you get the memory cells. And those memory cells are what are responsible for that secondary response. As you can see in the graph, the secondary response is much stronger, much more rapid, and much more long-lasting. And basically, because you have the memory cells, your body can respond much more quickly and much more strongly to the same antigen if you get exposed to it again. This is why once you get exposed to a particular pathogen, either through just the natural course of your life or through vaccination or immunizations, you don't get sick if you get exposed to the same pathogen. Most people get chickenpox. They don't get it a second time because now they have the memory cells. When you get a vaccine, you get something that will cause a primary response that won't actually cause you to become sick, so that if you actually got exposed to the actual pathogen, it wouldn't cause the serious side effects or possibly even death that might result, because now you can have this very strong secondary response, so you don't ever get the symptoms of the disease, or if you do, it's very, very short-lived and very minor. The last thing I want to talk about in this video are the different ways in which we can develop immunity. And there's 4 of them, as you can see in table 16.9 here. There's naturally acquired active immunity. This is what happens when you're living your life and you get exposed to some live pathogen and you have a primary response. You get the symptoms of the disease and your body fights it off, and now you're protected against getting that same particular pathogen again, at least getting sick from it. Artificially acquired active immunity is a vaccine. You get a weakened or a dead form, or maybe just a piece of the pathogen. Your body creates the memory cells. You don't get sick, and now you have developed this immunity to this particular pathogen. And then there are passive immunities, where your body doesn't make the antibodies, they come from some other source. Artificially acquired passive immunity is getting a shot of gamma globulins or antibodies, or maybe an antitoxin, like if you get bitten by a rattlesnake, for instance, they give you antitoxin, which is antibodies to the snake venom. So this is short term. You don't develop any kind of immune response on your own. The rabies shots would be another example of this. And then that's artificially acquired passive immunity. Naturally acquired passive immunity is where antibodies are passed from mom to either the fetus or to her newborn baby. And there's two ways that can happen. The antibodies can pass across the placenta, and so any antibodies that mom has made, during that pregnancy, anything that she might have gotten exposed to, now the baby has some short-term immunity to that, and that's important because the immune system of that baby isn't really going to be functioning very well until it develops further in life. The other way that this can happen is when mom breastfeeds, there's IgA antibodies in the breast milk that can pass from mom to the baby as well, and that's one of the benefits of breastfeeding.